So, Raat lecture was about root, Dicot root or या मानक root that was discussed in detail. In structure parts of Dicot root and Dicot here Manakot root. The topic is that is TSR Dicot stem. Generally is a stem is true. Agar stem is transverse sections at always the path is under microscope. At means tissues shows. The shortest reference TSR stem on या TSR dicot stem. In a transverse section of dicot stem, the atmosphere tissues parts. Well, this plant was dicot plant. Yeah, primary growth has been secondary growth has been then patan shoot ya stem is true. Ye karakam a transverse section very thin. That is under microscope was examined. So then patan different types of tissues as which under microscope shows here dicot stem, transverse section, the type of tissues in it. So first thing which was one of the first tissues on Bosnia, the first tissue of this dicot stem, that is outer protective covering, that is called as epidermis. And one tissue is on choice at dicot stem that is called as epidermis. The epidermis is true. You should ask as for what it is outer protective covering. It is outer protective covering and the stem also trust stem says she cover current. Number of numbers using a cell they are a single layer of cells. In cells, they are compactly arranged. The cells are compactly arranged. Compactly arranged. In atmosphere, cell the intercellular space is absent. Intercellular space is absent. And even cell assumption, even cell darmiyan space assumption. So that is why they are compactly arranged. Yeah, though even we can see that intercellular space here is absent. So usually epidermis assumption maybe waxy cover. There is a waxy cover outside of this epidermis. Usually waxy cover after waxy cover is called as cuticle. And there is outer waxy cover. And outside epidermis waxy cover, that is called as cuticle. It is called as cuticle. The cuticle is true. Which function it reduces water loss. Stem epidermis stomata present. In some plants, the stem epidermis stem has stomata also. In the case of stomata present. Awesome. But as a transpiration, usually cuticle awesome true. You should pay cover awesome waxy cover, so it reduces water loss during transpiration. The base can usually stem awesome true, epidermis is awesome true. Cancer plant multicellular hair-like structures. And stem hair stem hairs. Yeah, these are also called as trichomes. And usually epidermis awesome true. At epidermis multicellular hair-like structures, those are called as stem hairs. Trichomes. In trichomes, sometimes they are secretory in function. Yeah, sometimes they give protection from enemies also. So these are called as trichomes, yeah, stem hairs. Rootus muscle hairs, root hair one. The difference is that they are unicellular and these are multicellular. Rootus muscle hairs, root hair is unicellular, multicellular. Types can be on absorption of water minerals protection clear. So this is called as epidermis. And potential is using a Dicot stem epidermis is back layer of cells. Yeah, another tissue. That tissue is called as hypodermis. Hypodermis. Hypo means below, dermis means skin. Yeah, epidermis beneath. Yeah, beneath the epidermis or below the epidermis. The tissue layer of cells that is called as hypodermis. The hypodermis is true. It is made up of, it is colon chimeras in nature. Colon chimeras. It is colon chimeras. So, agar yaad aise, colon chimera asan cell wall asan colon chimera, cell. Yim wall of colon chimera asan, deposition of other cutengus and suburin, yim banan thick. Yim thick banan, at that time they give mechanical strength to plant. Whereas women are hypodermis. Now it is made up of colon chimera cells, colon chimera nature. So it gives mechanical strength. It gives mechanical strength. It gives mechanical strength. Usually hypodermis colon chimeras in nature. It gives mechanical strength in plants. Yes, and but multi-layer of cells, another tissue made up of multi-layer of tissue. These cells, they are parenchymatous. And hypodermis, after hypodermis, there is another tissue that is multi-layer tissue. Yes, it's that is called as cortex. It is called as cortex. Hypodermis pattern layer of cell tissue is a multi-layer the cells are parenchymatous. The cells are parenchymatous. Yeah, they are made up of parenchyma. This is called as cortex. And the layer of tissue that is single layer of cells called as endodermis. In a back of tissue that is single layer of cells, that is called as endodermis. This tissue is called as endodermis. This is the endodermis inner wall of the cortex. In the cortex wall the radial or tangential wall. So the endodermis in the can see cell tangential or radial walls. Number one, tangential wall and radial wall. Radial wall of cell, they are deposited. They get deposited by suburin. And even percentage deposition of suburin, they become thick. Endodermis barrel-shaped cells. They are barrel-shaped objective entrance. The shape of endodermal cells are barrel-shaped. They are barrel-shaped. So, in tangential or radial wall, deposition of suburin, thick. Band-like structures or strip-like structures. Strip or band-like structure. They are called as Caspin strips. They are called as caspine strips. Caspine strips. So, usually endodermis it gives also protection. And is dark tissue multi-layer, that is called as pericycle. Another tissue is called as pericycle. The pericycle it is made up of cyclorenchymous cells. Cyclorenchymous cells in nature. So it also uses mechanical strength to stem. The unbalanced situation is there is in the center there is a vascular tissue. It's a back of there is a vascular tissue. Usually vascular tissue too. Vascular tissue. The vascular tissue is made up of xylem and phloem. It is made up of xylem and phloem. The usually xylem phloem lie side by side and they make a bundle-like structure. That is one is called as vascular bundle. Which you must suppose just like this. Dicot's voice. Dicot means vascular bundles are in the form of a ring. एक objective entrance the pattern of vascular bundle in dicots. Pericycle. The pattern of vascular bundle in dicot stem. You show us a ring pattern. It forms a ring-like structure. It is a characteristic feature of dicot stem. Usually vascular bundle in dicots they make a ring-like structure. So this is the characteristic feature of vascular bundle in dicots. So you can have an entrance specific point. Yes. The xylem, suppose towards epidermis it is phloem. The inner side is xylem. Okay. The usually vascular bundle vascular bundle is in the sheath.
Yes, sir.
The vascular bundle contains a sheath outside in the big protective covering. A sheath that is cyclorenchymous. And usually a vascular bundle after the sheath, so sheath cyclorenchymous. So it was mechanical strength to it.
Yes,
The vascular bundle is in dicot true. You should ask cambium present. Vascular bundle. Outside more phloem, inner side more xylem. So xylem is phloem is darmiyan meristematic tissue that is called as cambium. The cambium is dicot. The cambium is present only in dicots and it is absent in monocots. Go cambium secondary growth gasan. Secondary growth it is because of cambium. The bunyadis pattern here cambium present the vascular bundle in dicots is open. The vascular bundle in dicot is open. It is open because due to the presence of cambium. Agar is monocotton misal monocotton cambium it is absent. So that vascular bundle is closed vascular bundle. You should neatest even objective given the vascular bundle in monocots is closed. In cambium absent. The vascular bundle is closed. It is present in monocots. But in dicots it is called as open. Why? Cambium is present between the elements. Cambium is secondary growth. What is the vascular bundle in dicots? Number one, it is a ring pattern. Number two, the vascular bundle is open due to the presence of cambium. Clear? Basic feature. Usually vascular bundle true. Vascular bundle vascular bundle. Now there are vascular bundles which are xylem and phloem. So usually xylem for example this is xylem. This xylem should be in stages. So in stages. So usually godness xylem by first formed xylem is called as protoxylem. Protoxylem. You can note the point. The first formed xylem is called as protoxylem. The protoxylem part is xylem, that is called as metaxylem. Metaxylem. What is the one is? Xylem can be classified into two types. One is called as protoxylem PX. Second is called as metaxylem. The protoxylem क्या हो? The first formed xylem. The metaxylem क्या हो? The after protoxylem the xylem formed, that is called as metaxylem. तो ये metaxylem या protoxylem. अमिच presence. अमिस कम ये vascular bundle is classify करन. अगर ये प्रोटोजाइलम अंदर की तो मेटाजाइलम नेबर की तथ कंडीशन एंड आर कंडीशन then protoxylem PX protoxylem is inside and metaxylem outside. The condition is called as and our condition. You choose exams as objective. So you shall present in stem. Go in stem, the vascular bundle has and our condition. Yeah, the protoxylem is in and our condition. Go in stem, the vascular bundle is in and our condition. That is protoxylem, that is in inside, the metaxylem is outside. The mother in the root in roots, the condition is reverse. In roots, the condition is reverse. That means that क्या शासन काम अंदर की इसी शासन metaxylem, नेबी की इसी शासन protoxylem. वो in roots, the protoxylem is outwards, outside, metaxylem is inside. Time साथ में तथ condition माने exar condition. That is called as exar condition. EXARCH. When protoxylem is outside, metaxylem is inside, this condition is called as exar condition. It is found in case of roots. The andar condition is found in root of stem. The exar condition is found in root of stem. वो exar condition is rootus में the andar condition is stem में छठी ये क्या फीचर तो हम पता नहीं is usually sinus में there is presence of pith. Beakha tissue that is called as pith, and in the center there is presence of another tissue that is called as pith. Usually pith tissue, it is made up of parenchyma cells. Yeah, parenchyma. It is made up of parenchyma cells. Parenchyma. Yeah, so the pith is parenchymatous in nature. The pith is also called as medulla. The pith is also called as medulla. So, वो संकस में क्या है pith? ये xylem या ये vascular bundle ये मन दरमियान शासन ये pith present touches on pith rays. They are called as pith rays या medullary rays. Pith rays are also called as medullary rays. ठीक? वो pith is on centers में जाए यानी parenchymatous tissues on centers में जाए is called as pith. Is on ये बार the form of is या lateral side में आए it is called as pith rays या medullary rays. And its function क्या है? Usually pith. It is used for storage purposes. Pith is used for storage purposes. Even pith rays या medullary rays, it helps in lateral conduction of water and minerals. Yeah. Usually medullary rays या pith rays, they help in conduction of lateral conduction. Our whole carbon from अगर suppose this plant, plant is water absorption get through root, is on leaf down. That is called as ascent of sac. Mother, you should side on the partner, left side or right side. What touches on lateral conduction? Well, lateral conduction further we have there medullary rays, yeah, pith rays. So they help in lateral conduction of water and minerals. Okay? So this is the structure of this dicot. One monocot. Monocot, dicot are the same, but much differences. There are some differences. Use a monocot. What do we find? Monocot is true vascular. Use an epidermis. Epidermis like that. Magar ta cuticle and in monocot, cuticle is absent. In monocot, cuticle is absent. Number 1 point. Number 2, epidermis, hypodermis, cortex, endodermis, pericycle, and etc. That means usually ground tissue well differentiated. In monocots, there is no well differentiation of ground tissue. There is no differentiation of well differentiation of ground tissue. Ground tissue, ground tissue, tissue, epidermis bone up to vascular tissue, and hypodermis, endodermis, pericycle, pith, medulla, etc. Human side is this is called as ground tissue. So yeah, well differentiated. Dicot. But in monocot there is no well differentiation of dicot, this ground tissue. That means you can differentiate numbers. Number point difference. Yes, one way usually show. Dicot is an arrangement of vascular bundles. That is in a ring pattern.
Yes, sir.
But in monocots, the vascular bundles are scattered. Suppose I give more stem, TS. Dicot is in this ring pattern. And a ring formation. In dicot is a ring formation. Other than monocot in suppose monocot is scattered. And in monocot, the vascular bundles are scattered. Different patterns ring pattern. Scattered. They are actually differentiation ground tissue. There's a vascular tissue, vascular bundles, they are scattered in this ground tissue, in this stem. This is the difference. Number 4 point. Usually vascular bundle, BB, vascular bundle. In dicot, it is closed. But in monocot, in dicot, it is open. In dicots, vascular bundle is open. But in monocots, it is closed. Here it is closed. Go inside closed rice. There will be no secondary growth. Back point differentiation. There is no secondary growth. In monocots, there is no secondary growth, but in dicots, there is secondary growth. So this is the difference between board questions exam. What is difference between monocot stem and dicot stem? Point cuticle is absent in monocot, dicot is present. In dicots there is well differentiated of ground tissue. In monocot there is no well differentiation of dicot. Third point, vascular tissue in the vascular bundle in dicot it is ring pattern, but in monocot it is scattered. Third, the vascular bundle is closed in monocot but open in dicot. So epidermis is absent in monocot, it is present in dicot. So secondary growth will be present in dicot but absent in. So this is all about structure of dicot and monocot and difference between monocot and dicot system. Thank you.